Welcome to this session on the pathophysiology of pain. I'm Brian Wainger. I'm a physician scientist at Massachusetts General Hospital. My research interests are on both motor and sensory neuron disorders. And as I said, I'm going to speak today about pain. Thank you to the organizers for inviting me to speak with you. These are the disclosures that I'd like to present before beginning. So today we have two goals. One is to discuss a broad review of pain anatomy and pathophysiology. And the second is to introduce some recent clinically relevant research highlights in pain. And I'll focus on three topics. One is channelopathy, another is using human nocicptors for disease modeling, and the third is connections between the immune system and pain. So I'll begin by distinguishing baseline pain, which is also known as nociceptive pain, and enhanced pain. And baseline or nociceptive pain is refers to pain resulting from a high threshold noxious stimulus. And in this graph here on the x-axis, you can see the sensory stimulus strength intensity, and on the y-axis is pain response. And so at baseline, there needs to be some magnitude of the noxious stimulus in order to elicit a painful response. And as the magnitude of that stimulus increases further, there's an increase in the size of the pain response. In enhanced pain, there can be an increased pain response to what is ordinarily a noxious stimulus, and that's known as hyperalgesia. There can also be a noxious response to what is normally an innocuous stimulus, and that's known as allodynia. And finally, there can be spontaneous pain, which is pain that is occurring without an actual stimulus. And I should say that enhanced pain can serve either a protective or a pathologic role. So for example, after a sunburn, one experiences a painful response to lukewarm water. And that response, as I said, serves a protective role by preventing further tissue injury. You're not going to do something like turn the water too hot, or you're going to be less likely to go out into the sun and worsen your sunburn. So that serves a protective role. However, after nerve injury, which could occur, for example, with surgery or with any other type of injury, a minority of patients develop pathological enhanced pain, and that pain can become chronic. And allodynia, hyperalgesia, and spontaneous pain are typical features of chronic pathological pain. So we can talk a little bit more about pain ontology, which are these different groups of pain. And I should say that how these terms are used can reflect either a research or lab-based environment and a clinical environment. And sometimes the terms can be used differently in those two environments, and I'll discuss that a little bit. But in general, as I said, nociceptive pain refers to an ongoing high threshold pain that ends as soon as the stimulation is stopped. So in this cartoon here, you can see that when there's a noxious stimulus, there's a response. When the noxious stimulus ends, the response is over. There's no response to an innocuous stimulus or a spontaneous pain. In inflammatory pain, there can be an augmentation of these responses. So there can be hyperalgesia in response to a noxious stimulus. There can be allodynia in response to an innocuous stimulus. And generally, there isn't as much spontaneous pain with inflammatory pain conditions. Third, there's neuropathic pain, and this is reflected by hyperalgesia, allodynia, and spontaneous pain. And as I said, in the lab, these terms can be quite clear in that inflammatory pain refers to pain in an animal following injection of a cocktail often that activates the immune system. whereas neuropathic pain refers to the pain behaviors in an animal following nerve injury. In patients, these terms are used, I would say, less concretely, although there are, of course, formal definitions for neuropathic pain, which are based on the dysfunction of the nervous system. So there can be decreased sensation in a particular region. There could, depending on what nerve is injured, there could be a motor response as well. But it really has to do with the quality of the pain, and that there's a burning or lancinating or electric quality to the pain. Okay, so let's turn now to pain anatomy, and I would highlight, as in our discussion over the next few slides, the idea of parallel processing and also convergent processing. So the first order pain sensing neurons, or nocicptors, have their terminals in the periphery in the skin. They have their cell bodies in the dorsal root ganglia just outside the spinal cord, and then they project to the dorsal horn of the spinal cord. The pain information ascends via two parallel pathways: the lateral spinothalamic pathway that projects to the thalamus and then to primary somatosensory cortex, and that pathway conveys information about the location and intensity of painful stimuli. There's also the spinoparabrachial pathway, and this pathway projects to limbic centers, including the amygdala, and serves to provide information about the affective components of pain, and that refers more to the hurt that accompanies pain or suffering. Looking in more detail at the different types of sensory neurons and where they project into the dorsal horn, nociceptors are broadly defined as unmyelinated C fibers. These can either be peptidergic, so they contain neuropeptides such as CGRP and substance P, or they can be non-peptidergic, and these could express, for example, the MERG family of G protein coupled receptors. There are also A delta fibers that are thinly myelinated. In contrast, A beta fibers and larger fibers like proprioceptors at baseline don't play a role in pain. And what you can see is these different fiber types project to different regions in the dorsal spinal cord. So for example, the peptidergic C fibers project primarily to lamina 2, whereas the non-peptidergic C fibers project somewhat deeper into the inner lamina 2. And A delta fibers project to lamina 1 and also the deeper lamina 5 layers. At baseline, the A beta myelinated fibers that are involved in multiple sensory modalities, including light touch, project deeply to this group of PKC gamma neurons, as well as to lamina 5. And what we're going to see is in the setting of the transition to chronic pain, there are many changes that occur in the wiring of the different neurons and the way they signal. And so that's what we'll try to understand. But I would say for now, you can note this idea of some pathways that are parallel and also some pathways that are convergent. For example, the dual input into the layer 5 neurons here. So our understanding of these first order pain sensing neurons called nociceptors has evolved a lot from the times of Descartes in 1664, where the idea of pain pathways meant there was a noxious stimulus in the foot that would pull a rope and elicit a pain response on the other end of the body. This has evolved in 1906. Charles Sherrington was the first to coin the term nociceptors, And within the past 20 or so years, there's been a rapid and expansive improvement of understanding of the molecules and receptors involved in pain. And so these include specific inotropic receptors on the pain-sensing neurons and specific ion channels. So many of the receptors are in the TRP channel family or transient receptor potential family, and these include TRPV1, which is the capsaicin receptor. That's the active ingredient in hot chili peppers. It can respond, in addition to capsaicin, to extremes of pH and heat. There's the menthol receptor, TRPM8, the receptor that responds to mustard oil and several other noxious compounds, TRPA1. And this here is the Japanese fugu pufferfish, which makes the toxin called tetrodotoxin. It's actually a delicacy in Japan because if it's prepared properly, there's apparently, I haven't actually tasted it, but there's just a hint of the toxin, and that produces some tingling on one's tongue when one eats it. However, if it's prepared improperly and there's too much of the toxin, it's fatal. Why do I mention this? One key property of these nociceptor neurons is they express voltage-gated sodium channels that are actually resistant to tetrodotoxin. These are some of the only channels in the body that have that property. And there's been a lot that's been done to characterize those channels and also to understand why evolutionarily it's important that this population of sodium channels is present. So despite the understanding of these individual channels, it actually is much more complicated, and some of this has been revealed within the past few years. This shows that although TRPV1, and the capsaicin receptor, is the main receptor for noxious heat, there's actually several other receptors that contribute as well, and this includes TRPM3, which I didn't mention, and TRPA1, which I mentioned as the mustard oil receptor. It can also respond to oxidative stressors as well. And so this shows, this again emphasizes this idea of convergence that can be occurring on the molecular level. But there's also examples of divergence. And so here's an example where the same receptor, this is a sphingosine receptor, is present in two different cell types, in neurons that are involved in itch signaling and in pain signaling. And the way that this receptor is coupled to intracellular signaling cascades is different in these two different cell types, and that's part of what gives these two different cells the divergence of function, despite the fact that the initiation of these signals is coming from the same receptor. So I thought that's kind of neat. So the first step in pain augmentation, I would say, is peripheral sensitization. And what this refers to is a reduction in nociceptor threshold for activation and an amplification of that signal. It's primarily restricted to an injury site and primarily involves heat hypersensitivity. And so in the setting of tissue injury, there's release of cytokines and chemokines. These activate inflammatory cells that release their cargo and activate nociceptors. And this activation really occurs through the activation of G protein coupled receptors that lead to intracellular signaling cascades that modify the ion channels and receptors that I talked about. So for example, signaling through prostaglandin E2 leads to phosphorylation of TRPV1, and that phosphorylation allows the channel to be activated at less hot temperatures. These mechanisms have been a component of clinical development interest. So for example, the antibodies to NGF, which have been investigated in arthritis treatment, are based on this idea of minimizing peripheral sensitization. But again, I should say that peripheral sensitization is largely occurs through phosphorylation of proteins, of existing proteins, and through modification of trafficking of proteins. And to a lesser extent, involves transcriptional changes in the synthesis of new proteins. In contrast, central sensitization, which refers to changes that are occurring in the central nervous system to augment pain signaling, are much more profound in that they occur on the level of transcription, making new RNA, and translation, making new proteins. And this shows a cartoon from the pioneering work of Clifford Wolf in the 1980s that really described this process. And I'll just take you through this. So at baseline, before a stimulus that induces central sensitization, in this case mustard oil, one can stimulate different areas of an animal's paw and then record in this case it's a motor output. And you can see that that with stimulation of this low probability firing fringe region, the stimulation is indicated with the red bar. There's an increase in the output response. However, after mustard oil treatment, so now this is in the setting of this early central sensitization, there's not only a marked increase in the firing when a different region that at baseline there's not only a marked increase in the firing within that region, but if you go to a separate region that initially was outside of the firing range of that particular neuron, there's now marked activation. So one of the core ideas of central sensitization is that there's an expansion of the affected region, and that hypersensitivity away from the site of injury is sometimes referred to as secondary hyperalgesia. And so what we're going to talk about now is the different the different mechanisms that underlie central sensitization. So one of the ways that this occurs is through summation and wind-up. So summation refers to a clinical phenomenon where with repeated stimulation of what initially might be an innocuous stimulus, there's an increase in one's pain perception through that repeated stimulus. Molecularly, this is known as wind-up, and these little dots here indicate stimulation of a peripheral neuron, a first-order neuron, and the electrophysiological trace, the action potentials here are coming from recordings of the second-order neuron. And what you can see is that with repeated peripheral stimulations, there's an increased response, initially not firing action potentials, and then consistently firing single action potentials, and then more action potentials. And then you see that the duration of this response in the second-order neuron actually outlives that of the stimulation. And molecularly, this is thought to reflect release of the NMDA receptor, which is a type of glutamate receptor, from block by intracellular magnesium and polyamines, so that with prolonged stimulation, these blockers of the pore are removed, and there's free flux of cations, sodium and calcium, into the cell. And these the entry of calcium in particular is what drives the transcriptional changes that occur that alter neuronal plasticity in a way that promotes the pain process and pain signaling. This can be reflected in the strengthening of individual synapses. So this synapse at baseline could be strengthened postsynaptically by an increase in the production of, say, glutamate receptors. It can be strengthened presynaptically through an increase in the transmitter release. Looking sort of even finer, this can occur through formation of new spines for synaptic transmission, whereby transmitters were released in the presynaptic neuron and received in the postsynaptic neuron. And this can occur to promote the formation of new synapses. And this is thought to be the mechanism of allodynia, whereby at baseline, these low threshold mechanoreceptors serve a function to mediate light touch. Whereas the nociceptors, of course, respond to noxious stimuli and mediate pain. And so what is thought to happen is there's a sprouting of new synapses from these low threshold mechanoreceptors into the superficial dorsal horn. And those connections are what drive allodynia, because now these low threshold mechanoreceptors that may be involved in light touch, that are involved in light touch, now are able to transmit their activation signals to the second order pain neurons and activate them, and that results in allodynia. Looking at this in another way, as I said, there's the low threshold mechanoreceptors invade the dorsal horn, and their connections then become wired to the second order transmission neurons that convey information about increased pain to the brain. Another mechanism by which this occurs is loss of inhibition. So there are inhibitory interneurons that damp down the activation of these second order transmission neurons and so the result of the activation of these neurons is to reduce pain signaling. One of the things that's thought to happen with central sensitization is that these neurons become either non-functional or they can actually die, and that serves further to augment this pro-pain signal that's coming from the transmission neurons. There are changes that occur in the neuronal milieu from other cell types, in particular immune cells and glia. So this slide shows a marker in green of T cells, and on the side ipsilateral to a nerve injury model, there's a massive influx of T cells into the dorsal horn where these second order neurons are, as opposed to the contralateral side where there's very little. And that activation or that migration of immune cells into the dorsal spinal cord serves further to rev up the second order neurons and increase pain signaling. So as people probably know, there are descending systems that at baseline tend to inhibit pain, and that's actually one of the main ways in which opioid signaling works, through pathways in the periaqueductal gray and the rostroventral medulla, signaling to the spinal cord and inhibiting pain signaling to the brain, similar to the interneurons that I described previously. However, in the setting of central sensitization, those descending signals that are normally inhibitory can actually become facilitatory, and instead of inhibiting pain signals, promote it. And I'll give two more examples. One is in central sensitization and in chronic pathologic pain, there's actually changes in the way pain is encoded in the brain. So in a healthy person, one might expect to see functional imaging studies that show activation in response to a noxious stimulus in the thalamus and in primary sensory cortex. However, in chronic pain, the activation in response to either an innocuous stimulation or just spontaneous pain are quite different. And they're different in particular in that there's a broader region of activation, and that activation includes regions like the medial prefrontal cortex and limbic regions. And the activation in these other areas of the brain that are primarily involved in this affective component of pain that I mentioned earlier can be quite important in pain responses. And so this is an example of changes, molecular changes in the anterior cingulate cortex. So one of these limbic regions. And what was shown in this group that a particular kinase called protein kinase M zeta, what is increased in this region, the anterior cingulate cortex, following nerve injury. And the reason why this kinase was thought to be important is it's actually related to the kinases that are involved in learning and memory. And so there's a parallel between the development of chronic pain and the neuroplasticity, the changes in the wiring in the brain that occurs in response to that I guess occur in response to pain, but also mediate the prolongation and the chronification of pain, harbors sort of a striking resemblance to the plastic processes that Eric Kandel and others have described as being responsible for memory. And actually an inhibitor of that particular kinase, when focally injected into this area of the brain, reduces this mechanical allodynia and returns the pain thresholds back toward baseline. So in summary, I've told you about baseline or nociceptive pain versus enhanced pain, and I've described two examples, inflammatory and neuropathic pain. I've explained that pathologic pain is due to molecular and structural changes that occur in the central and peripheral nervous systems through central sensitization and peripheral sensitization. And I've explained that central sensitization in particular is mediated by several changes, and these include the new connections from A beta fibers that contribute to allodynia, the formation of new synapses, loss of inhibitory neurons, immune cell activation that promotes pain, loss of descending inhibition, and changes in how pain is encoded in the brain. And so we're going to turn now to a couple of other examples of different pain conditions. And the first I'm going to mention is channelopathy. And so this story started in 2006 when kids in there were rare cases of kids who did not experience pain, and they performed stunts like putting knives through their arms or walking on coals, and despite this did not experience pain. They were studied by neurologists who found that their sensory nervous systems were otherwise intact as determined by quantitative sensory testing. And genetic analysis of these patients found that they had two non-functioning copies of a particular sodium channel, SCN9A or NAV 1.7. And so this presented just an amazing opportunity for understanding how human genetics could be leveraged to try to think about new developments for pain treatment. And actually, more recently, there have been functional imaging studies in these patients. And one can see that, that indeed, in response to a noxious stimulus, these patients do not report a pain intensity above 0, whereas healthy controls do. And you can see using functional imaging that in the healthy controls, there's activation of expected regions in the brain in response to noxious stimuli, whereas this doesn't occur in those patients. Just after this time, there were activating mutations that worked in a dominant manner in the same ion channel, SCN9A or NaV1.7. And so this involves also rare cases of something called familial erythromelalgia. And this, as I said, this is a rare pain condition that's characterized by paroxysms of severe extreme pain, most often in the limbs, the feet, and arms, but also ears and nose sometimes. And so this sodium channel was then looked at in the laboratory as well. And this is really exciting work from Steve Waxman's group. And so at baseline, one can look at the threshold for action potential generation in nociceptors. And what one finds here, this is called the Rio base. One sees here that you need about 130 picoamps to make these neurons fire. However, in the mutant neuron, in the mutant neurons, you can see that there's firing with injection of only 65 picoamps. When the same injection stimulus is used, there's much more robust firing. And so this is indicating that nociceptor hyperexcitability or increased firing is a prominent component of pathological pain. And based on similar analyses in the congenital insensitivity to pain patients that I'll show you in a minute, it really drives this idea that blocking nociceptor excitability, perhaps particularly through NaV1.7, is an attractive clinical target. And this has been done through several modalities. This is an example of an antibody that's directed against that sodium channel. With increasing concentration of the antibodies, there's a reduction in pain response. And in addition to antibodies, this approach is being taken by many companies using small molecules as well as antisense oligonucleotides and gene therapies. So there are several examples of very rare diseases that are due to mutations in ion channels. So I mentioned inherited erythromelalgia, which is a gain of function mutation in NaV1.7. There's also paroxysmal extreme pain disorder, which is a similar gain of function mutation. The channelopathy associated insensitivity to pain, which is a loss of function in NaV1.7. And there's a few other examples, including familial hemiplegic migraine. However, what became clear is that there are more mild mutations in these same genes that are responsible for common diseases. So for example, there are more modest gain of function mutations in about 30% of small fiber neuropathy with minor alleles that reach a frequency of 10%. And similarly in a different sodium channel that also is expressed quite selectively in nociceptors, there are activating mutations that contribute to that painful neuropathy as well. So I'm going to move to human nociceptors. So one of the thoughts about failure to translate insight from mouse models to humans is that the nociceptors might be different. And so there's been some exciting work from several groups now that have taken postmortem human nociceptors and done these same types of patch clamp recordings and really shown that the human nociceptors behave quite similarly to the mouse nociceptors in that they respond to these different agonists like mustard oil and ATP activating purinergic receptors, as I described earlier. These human nociceptors also sensitize to inflammatory mediators as a model of peripheral sensitization. Human nociceptors can also be made using stem cell modeling, where you can have human patient skin biopsies or blood samples that are made into patient-specific induced pluripotent stem cells, and these can be differentiated in the lab into nociceptor neurons for mainly disease modeling in this case. In other diseases, this approach is used for cell therapy as well. More recently, there have been strategies of transcriptional factor expression for transdifferentiation from other cell types to nociceptors. This strategy of expressing different transcription factors in nociceptors is able to make neurons that express many of the typical markers of nociceptor neurons, and also captures the function of those nociceptors, including responding to different trip channel agonists like mustard oil and capsaicin and menthol. This is via calcium imaging. These nociceptors also capture other key properties of bona fide nociceptors, including the release of peptidergic neurotransmitters like CGRP, whereas neurons that are made to be non-nociceptor neurons using this is an abbreviation for different transcription factors in the reprogramming. These do not mediate CGRP release. This can be used for modeling different diseases like familial dysautonomia, which is a disease in which there's a progressive loss of nociceptors, and this can be captured using this stem cell-based modeling technique in which neurons made from the familial dysautonomia patients can show decreased outgrowth and decreased branching of their neurites. More recently, this has been looked at in stem cell-derived nociceptors for the NaV1.7 knockout, so the congenital insensitivity to pain phenotype, and indeed the excitability of these NaV1.7 knockout neurons is reduced compared to controls. The last topic that I'll just mention just briefly is the immune system in pain. There have been several exciting findings within the past several years. One of these includes the idea that there's direct activation of nociceptors by bacteria and bacterial products, and that is a way of causing both pain and neurogenic inflammation. Neurogenic inflammation meaning that the neurons, through release of neuropeptides themselves, but also through a back and forth facilitatory interaction with immune cells to release pro-inflammatory molecules. More recently, there's been evidence that the bacterial stimulation of neurons actually can shut off the immune system and contribute to necrotizing infection. And there's some ideas for how to interfere with that. So I'll mention two last things. These are related also related to the immune system in pain. There's evidence for microglial activation in response to tissue damage and nerve injury and augmentation of pain signaling through that. This can involve disinhibition of nociceptive signaling. It is controversial, but it may be that microglial activation by opioids may actually contribute to the paradoxical increased pain that can occur with opioid treatment, and this is known as opioid-induced hyperalgesia. And this idea of microglial and astrocyte activation has in the brain has also been validated with imaging studies in low back pain patients and controls, and seeing that there's greater activation using, in this case, using PET tracers. So I'll leave you with just a few final take-home points, and these are, again, that in baseline nociceptive pain, pain results from an ongoing high threshold stimulation. When that stimulation ends, the pain ends. Pain facilitation, which is an augmentation of pain, can be protective, but can also lead to chronic pathological pain. Chronic pain is due largely to changes in the central nervous system that enhance the pain signal. And there have been recent studies that have identified a broad range of channelopathies that lead to chronic pain, as I described. And finally, there are genetic and human approaches and study of immune and nociceptor interactions that have identified new directions for potential clinical target identification. And I think based on these, I'm quite optimistic that within the next 5 to 10 years, there will be a range of new analgesics that will be coming to clinical trials, and hopefully soon after that to patients. So thank you so much. Please don't hesitate to reach out with any questions. Thank you.